Hi everyone, Mr. Binder here. This is chapter 11, the nervous system. We spoke about the endocrine system in chapter 10, and now we are going to discuss how the nervous system complements the endocrine system. Again, what the nervous system is just a bunch of nerve cells carrying messages to the brain from the body, and then again, the brain is going to interpret those messages and then send out an actual response. And that could be to a variety of uh, different places, cells, organ systems, tissues, muscles, you name it. The nervous system has control over a lot of the things that our body will do in terms of function. So taking a look here, this is what we're going to discuss, the nervous system broken down into the peripheral and the central. Again, you can see that the central is broken down into the brain and spinal cord. That's as far as we'll go for the central nervous system. The peripheral nervous system is broken into the autonomic and the somatic nervous system. Again, we'll discuss what those mean. But the autonomic nervous system is broken down into the sympathetic and parasympathetic nervous system, which is also known as your fight and flight response, fight or flight response. Again, taking a look here, when we discuss homeostasis and negative feedback mechanisms, or what we spoke about was negative feedback loops. Basically, what our body does is detect stimulus from the environment, either internally or externally. And again, the examples I gave you was thermoregulation and osmoregulation. Well, our afferent system is responsible for relaying those messages to the brain via the spinal cord. Again, the spinal cord is like the highway. All of the nerve endings or the receptors will make its way to the spinal cord. And again, the spinal cord is like that huge wire, that big, huge line where it just sends everything up to the brain. So again, these sensory receptors will detect stimuli. Our afferent system is responsible for sending those messages to the brain via the spinal cord. Again, this is where we have the hypothalamus, which is a region of the brain, integrating the information that it receives. And once it integrates it, it will then send out a signal, which is via the efferent system, so back down the spinal cord and out to the body. Now we have two different types of systems here that the efferent system is responsible for controlling here or functioning. The somatic system, which is our skeletal muscles, and our autonomic system, which is broken into our fight or flight response, controlled by the sympathetic and parasympathetic divisions. They are responsible for organ function, gland function. And we'll discuss what each of these divisions are kind of responsible for. In terms of signaling a cell or basically taking a message from a sensory perspective and sending it up to the brain, again, the cells that are responsible for the nervous system are called neurons or neuronal cells. Okay, they're specialized, very specialized, because they serve as sending signals from these sensory receptors to the brain, and then again back down to the somatic nervous system or the autonomic nervous system. Again, when taking a look at a neuron, there's three different parts. There's a cell body, dendrites, and axons. Again, taking a look at the dendrites here, these serve as the sensory mechanism, sending signals down to the cell body where it will travel down the axon, again to these axon terminals. At the axon terminal, this is where we will actually see one neuron connecting to another neuron to send the message, to continue to send the message. So taking a look here, we see the cell body. The cell body contains the nucleus and most organelles. Again, what does the cell body of a neuron do? Synthesizes proteins. Neurotransmitters are proteins. Again, taking a look here, carbohydrates and lipids, all of the macromolecules that are needed for this specialized cell to function properly. Dendrites, they are responsible for receiving sensory information, sending information to the cell body. And again, axons, these are the long extensions from the cell body. Again, they carry signals away from the cell body to axon terminals. And again, this is where we will talk about the transmission of a signal from one neuron to the next. There's three different classes. There's the afferent neurons, interneurons, and efferent neurons. Again, taking a look here, afferent neurons are sensory. They only transmit signals when a stimulus is sensed or received. Okay, so again, these messages from the afferent neurons will be sent to the brain. Interneurons, these are the interneurons, these are found in the spinal cord. Again, they integrate information, they help to formulate a response, but basically these interneurons will be found in the spinal cord. Efferent neurons, again, these are your motor or the neurons that are responsible for cause and effect based off of the sensory information that afferent neurons send to the spinal cord and then again eventually the brain. The efferent neurons will carry the response either to the somatic nervous system, which is your muscles, or the autonomic nervous system, which is comprised of your organ systems and your glands. Neural signaling, again, this is the components that are involved with detecting stimulus, okay, again, integrating information and then hopefully a response to correct the change that was picked up on, okay, by the afferent neurons. Again, we see reception here. This is the detection of the stimulus. Transmission is the movement of that message along the axon up to the brain. Again, integration occurs in the brain. This is interpretation of a message. And the response is the effect. Now, the efferent system. The message is traveling down the efferent system to the somatic nervous system or the autonomic nervous system. Again, and then hopefully there's a response, whether there's some sort of hormone being released or whether there's a muscle being contracted or a specific organ now working harder to bring things back to normal. Again, this is the flow chart, I guess, that you would actually kind of follow in terms of the direction of a message. Okay, so we see a receptor here. Some sort of stimulus caused this receptor to send a message through the afferent neuron. The afferent neuron will end up, okay, into the dorsal ganglion. This is a cross section of a vertebra here, okay, which is part of the spinal cord. Here we see an interneuron. Interneurons reside in the spinal cord. Okay, so afferent neurons carry messages into the dorsal root of the vertebra, which we see as part of the spinal cord. One or more interneurons, again, will relay that message up the spinal cord to the brain. Again, unless it's a reflex arc, which we will talk about, but that message will be sent up to the brain, integrated. Message will be sent back down the spinal cord through the efferent neuron, or sorry, another interneuron. That interneuron will connect with your efferent neuron. That efferent neuron travels out the ventral root. Again, dorsal and ventral are just positioning of the spinal cord here. The efferent neuron will then take that signal to, in this example here, a muscle, which is part of your somatic nervous system. And we see, hopefully, a contraction of muscle here. A reflex arc is a little bit different because instead of the message going to the brain, we see afferent neuron here sending message to the spinal cord, but instead of the message going up to the brain, it eventually will, the decision to make a reaction will happen right here in the spinal cord, and it will end up not going to the brain. It will actually end up going to the muscle. So again, you might see the example here, a pin. If we put our foot or finger on a pin, we'll feel that prick and we'll want to remove our finger right away. Again, same with the patella test. Doctors might have done this patella test before. You sit on the desk, okay, and doctors hit your patella tendon and your knee just naturally just moves. Okay, that is known as a reflex arc. The brain is not involved with this, so all we see here is the afferent neuron sending a signal through to the interneuron. It doesn't travel up to the brain. Instead of going to the brain, it ends up connecting with the efferent neuron, traveling down to your somatic nervous system, in this case the muscle, and we see contraction. For neurons to function properly, they need a support system. And again, taking a look at here, there's a couple cells that help in the support of these neurons to transmit signal. Schwann cells. Again, Schwann cells, we see here, they're wrapped around the axon and they form what's known as myelin sheaths. Now, myelin sheaths act as an electrical insulator. which allows for the transmission of a signal to occur a lot quicker, okay, or with increased magnitude. The gaps in between the myelin sheath here are called nodes of Ranvier. These nodes are again gaps between Schwann cells, and it allows for the transmission of a signal to occur a lot faster. And then finally, glial cells. These glial cells you see here, which are wrapping themselves around the axons of specific neurons. Again, they remove dead neurons and they provide nutrition and support. And again, they are involved with an immune response. They'll remove dead pathogen. Again, you take a look at the cell body here. Nothing changes with the eukaryotic cell. We still see the organelles are involved with your normal eukaryotic cell, nucleus, mitochondria, Golgi apparatus. Again, all of the necessary organelles for a specific eukaryotic cell to function properly. And some of the things we discussed earlier is DNA replication, protein synthesis, all of the things, lipid synthesis, all of the things that are necessary for this specific nerve cell to function properly. Again, nerve signals, all right, they use ATP, right, to generate current. And what happens here is they communicate across a synapse. So nerve cells, they actually send signals to other nerve cells. Again, we see the axon terminal of one cell here, you see it here, attaching to the dendrite of another cell here. So again, when a message is traveling down the axon to the axon terminal, we see a transmission of a signal here from one neuron to another. That will occur, and again, the signal will travel down the nerve cell until it reaches another neuron. Again, this is the transmission from afferent to interneuron, and again to the efferent neuron system. Two sides to a synapse, so basically this is when one neuron is connected to another neuron. Again, we see the presynaptic cleft, which is the axon terminal of one neuron, and that is attached to the postsynaptic cleft, or the dendrite of another neuronal cell. There's two ways in which neurons can signal each other, either chemically or electrically. This example here is chemically, and we see neurotransmitters actually attaching to receptors of another neuron to continue that transmission of a signal. Again, a chemical signal. This is the example here. There is a little space in between the axon terminal and the dendrite of another neuron, and that gap there, okay, which is known as the synaptic cleft. Neurotransmitters are used here to kind of transmit that signal from that axon terminal to that dendrite. Again, in a chemical synapse, neurotransmitters are used to communicate between neurons. Electrical synapse, we see the one axon terminal is connected to the dendrite of another neuron here via gap junctions. Again, there is no synaptic cleft or gap in between these two. They are connected through gap junctions. The transmission of a signal here occurs very rapid, okay? And what happens here is the ions will flow continuously from the axon terminal here all the way through to the dendrite of another neuron. Again, the conduction of electrical signals by neurons, there's a difference in charge across the membrane of a neuron. Again, taking a look, difference of charge involves ions and some anions, and we're going to take a look at how an action potential is created by a neuron. And what we see here is there's a difference in ion concentration, positive ions on one side than there are on the other side. And again, what we're going to see here when we do describe an action potential is there will become a change of concentration of specific ions across the membrane here, which will change the overall charge of the inside of a neuron as opposed to the outside of a neuron. And this is the basis for nerve impulses. Again, taking a look, resting membrane potential. So when a neuron isn't even functioning, It's just waiting for a stimulus so it can send a signal up to the brain. What happens here is it's at resting membrane potential. So that resting membrane potential is about negative 70 millivolts. Again, the cell is polarized. So when a neuron is at resting membrane potential, there's a difference of sodium ions and potassium ions inside and outside the cell. So we see here that inside the cell, we only see a certain amount of sodium ions as opposed to outside. There's a greater concentration of sodium ions outside of the neuron than there is inside. When we take a look at potassium ions, we see the exact opposite. We see that there's more of a concentration of potassium ions inside of the neuron than there is outside. And again, we see that there are some anions here that are negatively charged. They cannot pass through the membrane. They're always inside of the axon here. Again, so when you take a look at a nerve at resting membrane potential, the inside of the cell is negatively charged. The outside of the cell is positively charged. Again, we are going to talk about specific ion voltage gated channels here. We see the sodium gated channel and we see a potassium gated channel. These will open and close in response to transmission of a signal, which we will discuss. Again, the sodium-potassium pump is used at the end of an action potential to restore charges inside and outside of the cell. So again, we will talk about all three of these voltage-gated channels. And again, the action potential is when a stimulus is created. So when stimulus is created and we see this threshold potential here be broken, again at about negative 50 millivolts, an action potential will occur and a transmission of a signal will be sent through the neuron. And again, it could be as little as a little touch on your skin. Okay, anything that a nerve cell can respond to and transmit a signal. Again, taking a look at neurons here, they conduct an electrical impulse. Okay? Basically what happens here is when there's a change in membrane potential on a nerve cell, there will be a transmission of a signal. And what does that mean? Well, if the inside of an axon is negatively charged, when a signal is transmitted, the inside will now become positive. And it's this change in charge that will allow for the transmission of a signal to occur. And there's 6 phases that we're going to talk about. And again, this will be on the final exam. You need to know all this. Resting state, threshold, depolarization, repolarization, the undershoot, and then back to resting state. Again, all of these occur hopefully in order for an action potential to actually go. Again, neurons follow the all or nothing principle. Let's say a stimulus caused a change in voltage here at about 10 millivolts. Well, it's the all or nothing principle. So when membrane potential changes about 10 millivolts, the action potential will not actually occur. It needs to be greater than about 20. At about 20 millivolts, we see here a change in 20 millivolts. We'll see an action potential actually occur. That's what the all or nothing principle means. You need to know this for the test. This is very, very important. Again, we see here that these are failed initiations of an action potential. But again, when the stimulus is strong enough to break that threshold, an action potential will occur. And what does that mean? Well, during resting state, basically it means the opening and closing of specific channels here. And the ones that I described already are your sodium-potassium channelways. And again, these are protein carriers. Okay, they're basically just channels. Again, when we discussed this back in chapter 2, they're the opening and closing that allows for the movement of sodium ions into the axon or the nerve cell, and again, potassium ions to leave the cell. Again, resting state here, what does that mean? Both sodium and potassium channels are closed. A threshold has cause and action potential, so stimulus occurs here. What happens? The opening of sodium channels, only sodium channels in this case. Again, the all or nothing principle, about negative 50 millivolts needs to occur. When that happens, more sodium channels are triggered, and an action potential occurs. So again, we see sodium channels opening, depolarization, sodium channels are open, potassium channels are closed. Well, that means that sodium channels are going to, or sodium ions will rush into the cell. There's a greater concentration of sodium outside of the cell than there is inside. So again, the movement of ions based on the properties of concentrations. Higher concentration to lower concentration. So sodium ions will rush into the cell, making the axon more positive. More positive. And that occurs here with the depolarization. Depolarization means that sodium channels are open. Sodium channels are open. Very important here. Potassium channels are closed. We get to the top here, and what happens? Repolarization. So again, repolarization, and this is actually occurring as the signal is moving down the axon towards the axon terminal. So what happens here? Depolarization, sodium channels are open. Now we see repolarization, sodium channels close, potassium channels open. So that means that there's a greater concentration of potassium inside the axon than there is outside. Well, when potassium channels are open, potassium ions will now leak out of the axon and into the interstitial fluid. Now, what does that mean? That means that the axon is becoming more negative. Becoming more negative. Basically, that axon has transmitted a signal from that specific area and it is moving in a direction towards the axon terminal. This is known as repolarization. Again, we see potassium channels remain open. This is caused by this refractory period here, or called undershoot. This undershoot is responsible for maintaining the magnitude and direction of a specific transmission. This doesn't allow for the transmission to actually work back to the cell body. That's why a signal always goes in one direction, down the axon to the axon terminal. It's because of this refractory period. Again, sodium channels remain opened, hyperpolarization, or this refractory period, the membrane potential falls below normal, and then what ends up happening is potassium channels will close, sodium channels are already closed, and again, what happens here to restore the concentrations of sodium ions and potassium ions across the membrane here, we see the function of the sodium-potassium pump with the help of ATP. Again, the sodium-potassium pump will pump sodium ions out of the axon back into the interstitial fluid, and again, with the as it pumps 3 sodium ions out, 2 potassium ions will pump back in, and again the sodium potassium pump will restore charge across the membrane, but again it will also restore the concentrations of potassium ions inside of the axon and sodium concentrations outside of the axon membrane. Again, the refractory period is just to ensure that the transmission of a signal will occur in one direction. In one direction, again, that direction is down the axon terminal, or down the axon hillock here to the axon terminals. Factors that affect potential. Again, the magnitude of this signal always remains the same. Okay, the greater the stimulus, the faster the action potential is, and the rate of conduction increases with diameter of axon. So, when an axon when its diameter is thicker, the conduction will increase. Myelinated axons. There are myelinated axons, and again, there are not myelinated axons. The difference between the two is that we see with myelinated axons a jumping here of transmission from node of Ranvier to node of Ranvier. So myelinated axons or myelinated neurons will actually, the signal will travel faster. Saltatory conduction means that there is a hopping of these action potentials in between nodes of Ranvier. Again, what happens here? Sodium-potassium channels are crowded into these nodes, and we see a quicker response of transmission of signal here. Again, speeds of 130 meters per second compared to about a meter per second in unmyelinated neurons. Basically, that was the action potential within a neuron itself. Now, when a neuron wants to talk to another neuron or communicate with another neuron, it needs to do this through a synapse. Okay, and there's two different types of synapses. There's electrical synapses and there's chemical synapses. Now, chemical synapses, again, use neurotransmitters, which is produced by the nerve cell itself. Okay. What happens here is transmission becomes delayed. Okay, it becomes delayed, but what happens is that axon terminal, okay, can receive hundreds of thousands of transmissions at the same time. Again, conduction across the chemical synapse. You need to label this on the final exam, all of these different terms here, and you need to explain the conduction of a chemical synapse. So what happens here? Neurotransmitters are stored in vesicles. When an action potential reaches the axon terminal here, there is a release of calcium into the cytosol. That calcium will trigger a protein which allows these vesicles to fuse to the membrane. Well, we all know that this is exocytosis. When those neurotransmitters are released, they will travel across the cleft into receptors of a dendrite of another neuron. They will attach to those receptors and transmit a signal. The role of neurotransmitters, again, they diffuse across the cleft here. They attach to receptors on the dendrite of another neuron. Okay, again, we see sodium and potassium channels opening here. This obviously causes stimulating or inhibiting effects. And these are some of the neurotransmitters here, okay, that are responsible for transmitting a signal across a cleft: acetylcholine, dopamine, norepinephrine, serotonin, GABA, and then endorphins. Again, endorphins, your natural pain. GABA, this helps with sleep and movement. Again, serotonin is sleep, hunger. Norepinephrine, which is your fight or flight response, your mood, arousal. Dopamine, this is again your mood, arousal. Acetylcholine is again muscle action, movement, memory. Taking a look at these, they are proteins, and again, they are involved in the transmission of a signal across a chemical synapse. That first part of the nervous system we just spoke on was how a signal is transmitted through neurons. Again, through the afferent system, again to the interneuron system, which would be your central nervous system, which we'll discuss right now, and then again to the efferent system. Okay, afferent means bringing things towards, and then efferent means bringing things away. So again, afferent system bringing signals to the brain, efferent system bringing signals away from the brain. The central nervous system, again, this is the brain and spinal cord. This central nervous system, again, manages the body's activity, integrates information that it receives from the afferent system, and again, sends out a response. And this is obviously for organ function, maybe for endocrine gland secretion, and again, for muscle control. To protect the brain and the spinal cord, we see meninges here. Meninges is just 3 layers of connecting tissue that surround and protect the brain and spinal cord. Again, cerebrospinal fluid. This cushions the brain and the spinal cord. Again, it provides nourishment and protects it from toxic substances. The cranial nerves, when you take a look at the brain, and in that assignment that I gave you, there are some cranial nerves that I would like you guys to learn about. There are about 12 of them. They originate from the brain and brainstem, and they will travel down the central nervous system and again out to the periphery, and they will control organ function, again brain function and muscle function. These cranial nerves will branch out and control everything that our body needs controlling by. Again, there's motor neurons, there's sensory neurons, and these cranial nerves can act as both. And again, the last slide here, the spinal cord. This is very important because it carries impulses from the afferent system to the brain, and then obviously from the brain to the peripheral nervous system. Again, the spinal cord contains the interneuron circuits, but it also controls the reflex arc here as well. Again, a reflex arc is messages that are not received by the brain. They are just received by the spinal cord and then sent back out to the efferent system. What do you need to know about a vertebra? Well, it is composed of gray matter, which is nerve cells. Again, nerve cell bodies and dendrites. White matter consists of myelinated axons. The dorsal root is the location where an incoming efferent or afferent neuron will run into. Ventral root is an outgoing efferent neuron. Again, there's 31 pairs of spinal nerves in the central or in the spinal cord. Cauda equina, you might have heard of this term before. Again, this is just a collection of spinal nerves that leaves the inferior of the spinal cord right around here. Okay. Again, please review these slides. I told you what was going to be on the final exam. You need to review them. Please ask me questions. And again, these are what's most important here is the action potential, but also the chemical synapse.